It's really amazing when you think about it. A new medicine goes all the way from a scientist's lab to your medicine cabinet. But there's so much that goes into that journey.
Absolutely, especially when you get to those phase 3 clinical trials.
Right. That's where things get incredibly intense. It's all about statistics.
You said it. It's not just about the numbers, though.
Yeah, it's about building this rock-solid case, statistically speaking, to make sure the drug really works and is safe for people to use.
Like building a statistical fortress.
Exactly. And that's what we're diving into today. In this deep dive, we're going to unpack some of the hardcore statistical methods they use in these phase 3 trials. It's how we figure out if a new treatment is really worth getting excited about and ultimately getting approved.
These are the analytical tools that separate the truly promising therapies from the rest.
We've got a lot of great material here, covering all sorts of aspects of drug development. Our mission today is to zoom in on those statistical must-haves for phase 3. It's all about statistical power and significance.
These concepts are crucial.
They really are. They can make or break a drug's chances long before it even has a chance to help someone like you or your loved ones.
Before we dive into the specifics, it's important to remember that phase 3 trials are where the rubber meets the road in drug development.
They're the big leagues.
Precisely.
So to kick things off, where should we start with all this statistical heavy lifting in phase 3?
Let's begin with this idea of statistical power. Imagine it like this. Power is the trial's ability to spot a real treatment effect, but only if a real effect actually exists.
So it's about seeing a real signal through all the noise and the data we get from these trials, right?
Exactly. Think of it as the trial's ability to pick out a genuine melody from a symphony of background sounds.
That's a great analogy. So if a drug truly works, a trial with strong statistical power is more likely to actually show us that it works. Makes sense.
Precisely.
What determines how much power a trial has? What factors play into that?
There are three main players: the number of participants in the trial, or the sample size, then there's the size of the treatment effect we're looking for, and of course there's the inherent variability in the data we gather from those participants.
Got it. More people means more reliable data, and a strong drug effect should be easier to spot, naturally.
Right.
But what about data variability? What's that all about, and how does it affect things?
So data variability is all about how spread out the measurements are within each group of patients in the trial.
Oh, I see.
If people respond very differently to the same treatment, that natural variation makes it tricky to pinpoint a consistent drug effect.
Because it's all over the place.
Right. Now, this ties into something we found in the Clinical Trials Handbook. It highlights a really important point. Smaller trials are more prone to imbalances between the groups when we use simple randomization.
Randomization just means we randomly assign patients to either get the new drug or a placebo or an existing treatment, right?
Exactly. But even with randomization, just by pure chance, one group might end up with more patients who have a worse form of the disease than the other group, and that could obscure a real benefit of the drug.
It's like the luck of the draw working against us, right?
Exactly. That imbalance can actually weaken our statistical power and make it harder to see if the drug is truly making a difference.
So in a small trial, the way we divide the patients into groups can have a big impact on the results, even before we factor in the drug itself.
Exactly. And the handbook points out that in larger trials, simple randomization might actually be enough. Seems like the size of the trial plays a huge role in shaping our statistical strategy from the very beginning.
Makes sense. The more people you have, the less likely you are to get those uneven groups just by chance.
Right.
Fascinating. What are those smaller trials where we might have to worry more about imbalances?
Well, the handbook talks about this technique called minimization. It's a more refined way of assigning participants to treatment groups, where you take into account those key baseline characteristics like their age, how severe their disease is, and other factors.
You make sure the groups are balanced from the get-go.
Exactly. That way, we can have more confidence that any differences we see are actually due to the drug, not just an accident who ended up in which group.
I get it. So it's not just about having enough people, but also making sure those people are divided fairly. Now, what about this significance threshold? That sounds a bit more abstract and statistical.
Yeah, the significance threshold, often represented by the Greek letter alpha, is kind of a decision-making tool. It helps us determine if the results we see in a trial are because of the treatment or just random chance.
So we need to be able to tell if what we're seeing is a genuine effect of the drug, not just a fluke.
Right. To understand this, we have to think about the hypotheses we're testing. In a typical phase 3 trial, we start with something called the null hypothesis. It's the assumption that there's no difference between the new treatment and whatever the control group is getting, either a placebo or the standard treatment.
So we start by assuming the drug doesn't do anything special. Then we see if the data convinces us otherwise.
Exactly. The alternative hypothesis is that there is a real, meaningful difference between the treatment and the control.
I see. So we gather all this data to see if we have enough evidence to reject that initial null hypothesis and say, hey, this drug actually works.
Exactly. Now, the significance threshold, usually set at a p-value of less than 0.05, comes into play here.
0.05. What's that all about?
It means there's less than a 5% chance that we'd see these results if the drug actually didn't work.
So it's like saying we're willing to accept a tiny risk of being wrong, but only a tiny risk.
Right. This is where we have to be really careful. Rejecting the null hypothesis when it's actually true is what we call a type I error, or a false positive. It means we'd be claiming the drug works when it really doesn't, and that can have serious consequences. One of the sources we looked at emphasized how important it is to test this null hypothesis rigorously to avoid those false positives.
It's like we're walking a tightrope between wanting to find new treatments and making sure we don't get fooled by random chance.
Exactly. And it gets even trickier. What if we set that threshold even lower, say to 0.01? We'd reduce the risk of a false positive.
So we'd be even more sure that the drug actually works if we see a positive result.
Yes, but here's the catch. Making the threshold stricter also makes it tougher to reject the null hypothesis, even if the drug has a real but small effect.
So we might miss a drug that actually helps people, just because we set the bar too high.
Exactly. That's called a type 2 error, or a false negative. And the likelihood of that happening is tied to the statistical power of the trial. The more power we have, the less likely we are to miss a real treatment effect.
So it's this constant balancing act, right? We don't want to give people false hope, but we also don't want to dismiss a potentially helpful treatment.
Precisely.
This really highlights how important it is to set those statistical parameters just right. It's not just about plugging numbers into a formula.
Right. There's a lot of careful consideration that goes into it.
Now, let's talk about the actual analysis of the data from phase 3 trials.
Right. Once a trial's up and running and the data starts pouring in.
What happens then?
Well, one crucial thing is that we have to decide how we're going to analyze the data before we even enroll a single patient.
We can't just wait and see what the data looks like and then choose the analysis that makes the drug look best. Right?
Exactly. That would be like changing the rules of a game in the middle of playing it. It wouldn't be fair, and the results wouldn't be reliable.
Right.
We need to lay out those rules in the study protocol up front. That's how we make sure the assessment of the drug's effects is fair and objective.
So it's like having a clear game plan before you step onto the field. No changing strategies mid-game. Okay, so what are some of those analysis techniques? What tools do statisticians use to analyze data from phase 3 trials?
Well, it depends on the kind of data we're looking at.
Of course.
If we're comparing something like blood pressure changes or scores on a questionnaire between two groups, we might use something called a t-test.
A t-test?
Yeah. One of the books we read, Clinical Trials, a practical guide, gives an example of a t-test in a slightly different context. They talk about comparing a group's average lung function to a value from medical literature.
I see.
But the basic idea is the same. In a phase 3 trial, we'd use a t-test to see if the average change in, say, lung function is significantly different between the group getting the new drug and the control group.
So a t-test tells us if there's a meaningful difference in the average outcome between two groups.
Exactly.
But what about when we're looking at events that happen over time, like how long patients survive or how long they stay healthy? We can't really use a t-test for that, right?
Yeah, you're right. For those kinds of things, we need different tools. In oncology trials, where survival is a major focus, we often use things like Kaplan-Meier curves and Cox proportional hazards models.
That sounds pretty complex.
They are, but they're powerful tools for understanding how a drug affects those crucial time-to-event outcomes.
So what do those tools tell us?
Well, Kaplan-Meier curves give us a visual representation of the survival probability over time for different groups.
So we can see if one group is doing better than another.
Exactly. It's a clear picture of how the treatment affects the disease progression. The Cox model is a bit more complicated. It estimates the hazard ratio, which tells us the relative risk of something like death or disease progression in one group compared to another.
And it takes into account other things that could be affecting the outcome.
Right. It helps us isolate the effect of the treatment.
That's impressive. Now, one of our sources also talks about non-inferiority trials. That's where you're not trying to prove a new drug is better than an existing one, right?
You got it.
It's more about showing it's not worse by a certain amount.
Yeah.
Why would we do that?
Well, sometimes a new drug might not be better at treating the disease itself, but it might have other advantages.
Like fewer side effects.
Exactly. Or maybe it's easier to take or cheaper to make. So in those cases, we're not aiming for superiority, just non-inferiority.
I see. So the goalposts are a bit different in those trials.
Precisely. Instead of trying to reject the null hypothesis of no difference, we're trying to reject the hypothesis that the new drug is actually worse than the old one by a certain margin.
That's interesting. It's like we're flipping the script on what we're trying to prove.
Exactly. It's a subtle but important distinction.
Absolutely. Now, what about confidence intervals? What are those, and how do they fit into all this?
Confidence intervals are basically a way of saying we're pretty sure the true effect of the drug lies somewhere within this range. One of our sources even gives a formula for calculating them.
Like a margin of error, right?
Exactly. It gives us a sense of how precise our estimate is. A narrow confidence interval means we're more certain about the true effect of the drug.
So it's not just a single number. It's a range that gives us more information about the results.
Right.
This is all so fascinating, but with all this complex statistical stuff going on, what are some of the challenges or potential pitfalls researchers need to watch out for?
Oh, there are definitely a few things to be careful about. One big one is multiple testing.
What's that?
Well, in many trials, researchers are looking at several different outcomes or doing lots of subgroup analyses.
Like seeing if the drug works better for men than women, or for older patients versus younger ones.
Exactly. But each time we do a test, there's a chance of getting a false positive result just by random chance.
Just because we're looking at so many things.
Right. So if we do too many tests without taking that into account, our overall risk of finding something significant that's actually just a fluke goes up.
Like if you flip a coin enough times, you're bound to get a bunch of heads in a row eventually, even though the odds are 50-50.
Exactly. So statisticians have ways of adjusting the significance threshold to account for multiple testing and control that overall risk of false positives.
So we don't get fooled by random noise in the data.
Right.
What other challenges are there?
Another big one is missing data. It's almost impossible to have complete data from every single patient in a trial.
People drop out, miss appointments, things happen.
Exactly. But if we don't handle that missing data properly, it can bias our results.
We're like trying to solve a puzzle with missing pieces.
Right. So we need to use sound statistical methods to account for those missing pieces and make sure our conclusions about the drug are accurate.
One of our sources, which focuses on biotech drugs, mentions something called target-mediated drug disposition. What's that, and could it create problems for the statistical analysis?
Yeah, target-mediated drug disposition is basically when the drug interacts with its target in a way that affects how it's absorbed, distributed, metabolized, and eliminated by the body.
Sounds complicated.
It can be. This can lead to nonlinear pharmacokinetics, meaning the relationship between the dose of the drug and how much of it ends up in the body isn't a simple straight line.
So it's not just a matter of more drug equals more effect.
Right.
So we need to use statistical methods that can handle that complexity.
Exactly.
And it all comes back to making sure those regulatory submissions like the IND and NDA are based on solid evidence.
Absolutely. Those applications rely heavily on the data from phase 3 trials.
And that data has to be rock solid, both in terms of how it was collected and how it was analyzed.
Right. It's all about ensuring that the drugs that eventually reach patients are truly safe and effective.
It's amazing to think about all the statistical scrutiny that goes into developing a new medicine. It's definitely not just a gut feeling or a promising early result. It's a data-driven process from start to finish, with statistics at the very heart of it.
Absolutely. Those statistical considerations, power, significance thresholds, and rigorous analysis techniques aren't just theoretical concepts. They have a real impact on whether a new treatment makes it to the market and can potentially help people.
As we wrap up this deep dive, it really makes you think about the crucial role statistics play in bringing new treatments to light.
It was not just about numbers.
It's about safeguarding the health and well-being of everyone who might benefit from these new therapies.
Exactly.
These statistical gates ensure that the medicines we rely on have been thoroughly vetted. And it makes you wonder, what would happen if those standards were lowered or compromised? It's a delicate balance between the need for new therapies and the importance of statistical certainty.
It's definitely something to ponder. And for anyone who wants to delve deeper into this fascinating world, I highly recommend exploring the fields of clinical trial design and biostatistics.
That's great advice. There's a whole world of knowledge out there for those who are curious.
Absolutely.
Well, this has been a truly insightful deep dive. Until next time.
It's been a pleasure.